Welcome back. We are starting chapter 41 and we are going to discuss the microbiology of the food in this chapter. So as we start, that first what we are going to talk about is the microbial growth and how it can cause the food spoilage. And we are going to talk about two different terms, which is the intrinsic and extrinsic factors which influence the food spoilage. And we are also going to explain the composition of a food which helps determine the kind of microbial growth that might occur. Like what kind of microbes grow if the food is carbohydrate, if it is a protein, if it is a fat. And we're going to talk about a couple of physical and biological features that influence the spoilage. So what exactly happened and what does it mean by food spoilage? So food spoilage results from growth of microbes on the food which are not desirable. It alters the food visibility and in other ways rendering it unsuitable for consumption. And it involves predictable succession of microbes. That means that the first group of organism that grows when the food is getting spoiled is different than the last batch of microbes that will spoil the food. So over the period of time, as the food is getting spoiled, the population of the microbes do vary from the beginning to the end. And different types of food undergo different types of spoilage process. Sometimes toxins are also produced by some of these microbes as well. So let's take a look at this, that the microbial growth is controlled by two factors. We talked about this. So the intrinsic factors are related to the food itself. First, we are going to talk about that. And then we are going to talk about the extrinsic factor, which is the environment where the food is stored. So what kind of intrinsic factors are in there? So for example, the fruits, many of them do contain this complex carbohydrate, which is the pectin, and then pectinolysis is breakdown of the pectin. When different microbes break down pectin, which is called the pectinolysis, it will produce methanol, then uronic acid, and this will lead to the soft rot and loss of the food structure. Now, it is very different in a meat, which mainly do contain the proteins, and this will lead, the microbes will lead to proteolysis and deamination and will produce certain types of chemicals in there, which are peptides, amines. They can produce hydrogen sulfide, ammonia, indole, which leads to bitterness, souring, bad odor, sliminess. These are some of the ways that the meat products or the protein-rich products are spoiled. On the other hand, the carbohydrates or the starchy foods also undergo the hydrolysis or the fermentation. These hydrolysis or the fermentation leads to production of organic acids. That means the pH will be dropped, production of carbon dioxide, and also there will be mixed type of alcohol. And these leads to souring, acidification of the material. On the other hand, the lipids like such a butter, mainly the spoilage includes the hydrolysis of the fatty acid and means that most of these fats are made up of fatty acid and glycerol. They are triglycerides. And the glycerol and the fatty acids are separated and by the process of hydrolysis. And this includes the rancidity and bitterness of the material or the lipid products. And we're going to talk about in detail that how different types of food do undergo these food spoilage. So let's take a look. So here what we are looking at, the first is the intrinsic factor of the carbohydrates. And you can see that on the left-hand side of the picture that this is basically a piece of bread. And we call this as a bread mold. And likely this blue greenish type of mold that is growing is Penicillium species. And this Penicillium is basically the genera from where penicillin was discovered. So this is a Penicillium species. Some of the Penicillium species is actually used in making some of the food products on the other hand. And we're going to talk about at the very end of the chapter when we talk about the cheese making. So basically, these molds will degrade because they basically are feeding on the carbohydrates that is available to them, which is the bread enriched in these carbohydrates. And little or no odor due to this. On the other hand, there is also another thing very common is the ergotism, which happens in the ear rot of the corn. And this produce an alkaloid, which is hallucinogenic, and sometimes it is a hepatotoxic. It is toxic to the liver, and this is released by a specific type of Ascomycetes, Claviceps purpurea produce these alkaloids, which is ergotism. These can lead to serious health concern. So after that, we are looking at the intrinsic factors of the proteins and the fats. And in this, mainly the bacterial growth predominates compared to the mold that we have seen in mainly carbohydrate or starchy foods. There are some specific words that we use. Putrefaction is a proteolysis and anaerobic breakdown of proteins to foul-smelling amine compound. And these foul-smelling amine compounds, there are two of them that has been shown here. One is called cadaverine, and you can see that the term comes from the cadaver, so you can well understand that what it is. It's cadaverine. The other one is putrescine. So these are these foul-smelling product. So unpasteurized milk, on the other hand, is spoiled by acid production followed by the putrefaction. So it is not that just only unpasteurized milk would undergo spoilage. Even pasteurized milk past its shelf life will undergo this kind of degradation. So there is a kind of a succession of microbes that happens in this process. So initially what happens that first, mainly the bacteria like Lactococcus lactis, which is subspecies lactis, or Lactobacillus is grown. These are mainly going to convert the lactose, which is the milk sugar, into the lactic acid. And then generally after the initial lactic acid fermentation, yeast and mold will start growing and they will degrade the lactic acid, and the acidity at the later part of that product will actually get degraded. And then finally there will be another group of bacteria which will digest the protein, and at that point it will start smelling, which is a putrid odor, and it will taste bitter. And then finally, the milk, which you can see right here, there will be a clearing of the milk with this coagulated fat and the protein, and they will gradually settle in the bottom. The consistent opacity of the milk will be gone, and you will see these flocculants of the coagulated milk and the protein. So this is basically the intrinsic factor, how the protein fat rich food can get degraded. On the other hand, there are many other intrinsic factors, other type of macromolecule type. Say, for example, the pH impacts makeup of microbial community and therefore types of chemical reactions that occur when microbes grow in food. Say, for example, the yeast and the mold will prefer growth in the low pH. On the other hand, at the neutral pH, the bacteria is more predominant, which can grow on the meat product as well. Another very important thing is the presence and availability of water. So the availability of the water in general is expressed as the water activity, or AW. We talked about that somewhere else. And in general, the lower water activity inhibits microbial growth. What we mean by that there is some moisture in the food, and whether that moisture or the water is available to the microbes in the environment which is close to that food product or not gives you the idea of water availability. Say, for an example, what happens that when you do the pickling process with a brine with acetic acid like vinegar, at that time the availability of the water goes down. On the other hand, the cooking also alters the oxidation reduction potential. So usually, if the lower redox means that the cooked food, which is the reducing environment after cooking the meat, say for an example, these amino acid, peptides, and the growth factors are released, means that some of the proteins are degraded, which is available in the form of a protein, amino acid, and growth factors. And these can lead to development or growth of different types of microbes, including, say, anaerobes like Clostridia, which can cause a serious health concern and food poisoning. On the other hand, the physical structure can also increase the surface area of the food. Say, for an example, the grinding, mixing distribute the microbes and promotes the microbial growth. So, for example, when we are just making sausages or the hamburgers, the chances of them getting spoiled are faster than solid meat product. So the outer skin of the vegetable and the fruits also slows the microbial growth. Say, for example, slight bruising of the tomatoes can lead to faster decomposition. So next, what we are looking at, there are many antimicrobial substances. Those are present in many of these Spices. So for an example, that in many of the herbs like sage or rosemary, we know that there are many of these antimicrobial species that grows in our herb garden. But there are some species like Bacillus cereus, Clostridium perfringens, sometimes Salmonella can be detected even in the spices as well. But what are these microbial substances? There are many different types. These coumarins are present in the fruit and vegetables, and these compounds are naturally antimicrobial. Lysozyme, on the other hand, is present in the eggs. So the lysozyme basically break down the peptidoglycan of the bacterial cell wall. We have talked about that before. So that's antimicrobial. On the other hand, in the cinnamon, mustard, oregano, they contain these aldehyde and phenolic compounds, and they also do have these antimicrobial activity. On the other hand, say these are some of the example: allicin in garlic, eugenol in the cloves, rosmarinic acid in basil, and then many of the polyphenols in the green and the black teas. These are all naturally occurring antimicrobial substances. Next, what we are talking about is the extrinsic factor. That means when the food are stored into an environment, temperature, relative humidity, and the atmospheric oxygen and carbon dioxide lead to how the shelf life of that food product will be. So for example, lower temperature slow the microbial growth, and this is very, very common that we all use refrigeration. In the household, we just use a 4 degree to 5 degrees Celsius, then minus 20 degrees Celsius. Then some of the industries that process food will also package the food and then transport them in refrigerated condition, which goes into the refrigeration room of the supermarkets. And even the shelves are refrigerated, or the cabinets are refrigerated as well. So you have to be very careful about that because what happens, say for example, the citrus products, which are not pasteurized, you just can have these concentrated citrus product that we see. They can be frozen, but upon thawing, they can really go bad pretty fast by the growth of yeast and the gram-positive bacteria. On the other hand, most of these frozen concentrates or the citrus concentrates are pasteurized, filtered, and concentrated by heating under the vacuum. You can easily see on the shelf of the supermarkets that there are many of the juices which is written as from concentrate. And usually these are the products which is pasteurized, have a longer shelf life than the unpasteurized one. On the relative humidity, higher levels promote microbial growth. Water availability, absolutely, that is what it is. And on the other hand, the atmospheric oxygen also promotes microbial growth. Some of the plastic films in which the food products are packaged can diffuse oxygen, and that can lead to surface microbes to grow. So excess carbon dioxide, on the other hand, can inhibit the microbial growth. So packaging of the meat products in high carbon dioxide concentration can lead or inhibit microbial growth as well. So let's take a look at the various methods that are used to control the food spoilage. And we're going to talk about the limitation of refrigeration, canning process, going to compare the pasteurization, what are those? Also, principle of water availability that we just talked about. And we're going to talk about the chemicals that is used in the preserved food and irradiated food, how the radiations are used, and also microbe-based food preservatives, where we used microbial products. And also, we are going to look at the packaging of these products to prevent the spoilage. So food preservation is a goal of food preservation is to eliminate or reduce the population of spoilage and disease-causing microbes while maintaining the food quality. So the microorganisms can be removed from the water, wine, beer, juices, soft drinks, and other liquids by filtration process. And this filtration process may better preserve the flavor and aroma than pasteurization, where you do have to heat the product to some extent. So usually this process could be really expensive because the filters that is being used can be clogged pretty quickly. So in the filtration process, initially the product is centrifuged. So the larger particles in these beer, juice, or soft drinks are precipitated, and then they are passed through a pre-filter. And once they pass through a pre-filter, they are filtered through a much finer filter, which filter out many of the disease-causing microorganisms. This prolongs the life of that finer filter, which are more expensive to produce. This better preserve the flavor aroma, as we already said. Then the Low temperature or refrigeration below 5 degrees Celsius slow the bacterial growth and it becomes bacteriostatic at that point, but it does not stop microbial growth. The microbes can still cause spoilage with extended storage. So you all know that when you just have the milk from the supermarket, usually there is a 2-week window that you can store your milk in the refrigerator. And after 2 weeks, it's going to get spoiled even in that refrigerated temperature. So even below 10 degrees Celsius, there are growth of microbes in fruit juice concentrates, in ice cream, and some fruits. Another very dangerous microorganism is the Listeria monocytogenes. And this Listeria monocytogenes, which gives you listeriosis, can grow at refrigeration temperatures and is very dangerous. So here we are looking at some of the usage of high temperature when we can food material. And this was first used by Nicholas Appert in 1809. He first introduced this canning process. So what it does, that the food is heated in a special container, which is called the retorts, to about 115 degrees Celsius for, depending upon the food, 25 to 100 minutes. This kills the spoilage of the microbes, but not necessarily all microbes. So they reduce the number of microbes to a safe level. But there are some of the problems. Sometimes what happens, the spoilage of canned food due to the spoilage before canning, under processing during canning, leakage of contaminated water into the cans during the cooling process, that can happen as well. So necessarily this process that we also do in home canning process, this is just a process much larger in the industry where once a soup or a vegetable is produced, they go into the can and they are heated to the certain temperature and then they are sealed into a condition so that nothing goes in there. But what happens that if you just get a can, and sometimes due to this contamination, there can be bacterial growth inside a sealed can as well. And it can produce organic acid, salt, and it can produce some of the gases. And sometimes you can see that if you just have a packaged canned food and somehow the can is bulging out, that is a clear indication that there is something growing into that can. So there are some of the processes that can lead to contamination even after the high temperature. Then what we are looking at is the pasteurization, and it kills the pathogen and reduces the number of spoilage organism. And there are different pasteurization procedure, heat for different lengths of time. Shorter heating times result in improved flavor because many of the proteins can quickly get denatured at higher temperature, and that can lead to some of the loss of a flavor. So let's take a look at what they are. The low temperature holding, which is called a batch processing. In this batch processing, the temperature is raised to 68.2 degrees Celsius and it is kept at 30 minutes. And usually beer, fruit juice, smaller volumes of milk that are produced in this way, which is called the batch processing. Whereas what you are seeing that a high temperature short time, or HTST, is the product is flash pasteurized for 15 seconds. The temperature is now 72 degrees Celsius. It's a little bit higher. And this is industry standard for milk and other dairy products in the United States. So what happens that there is a pasteurization chamber where the milk is kept at 4 degrees Celsius in the cooling condition, and it passes through a series of heat exchanger where through this heat exchanger, it is exposed to 72 degrees Celsius for 15 seconds, and then they are rapidly cooled down to 4 degrees Celsius back again before they can be bottled. So that's preserve most of the flavor of the milk without holding it for such a long time, which is the batch method. On the other hand, there is some other process that's the ultra high temperature, which is 138 degrees Celsius for 3 seconds, very short period of time. And this is also done by a very, very efficient heat exchanger. And the milk that does not require refrigeration until opened that you can get in many of the supermarkets, those are the ones which is pasteurized by this process. But it will have some loss of flavor, and usually most of them are flavored milk in that sense. So next, what we are looking at is the water availability. The water availability depends whether the microbes can go on the food or not. So usually say jam, jelly, pickle, these are some of the examples where the food can be stored for a longer period of time, even if they are rich in sugar and the substrate on which normally microbes will grow. So usually salt, sugar, vinegar, all of these are used, which will make the available environment hypertonic. Hypertonic environment is detrimental for the growth of many microbes. That means that they cannot grow because the availability of the water, which is called the water activity or AW, is reduced. So, but there are some examples that there are some bacteria we already know. They are known as the osmophiles or the xerophiles. These osmophiles and xerophiles still can grow in conditions in this hypertonic environment of these food, which can spoil these pickles, jam, jellies, as such. On the other hand, there is a process which is a dehydration, such as lyophilization to produce freeze-dried food is a common means preventing microbial growth. And this is probably one of the very, very foolproof method. So the freeze-dried food are basically quickly frozen, and under the frozen condition, a vacuum is applied. So when the food is frozen, a vacuum is applied. That process is known as lyophilization, which is a dehydration in the cold condition. So what is the advantage of that? So the food is frozen and all these, it is solidly frozen. At that time, when you apply the vacuum, the water directly from in the iced condition directly goes into the vapor condition. There is no liquid intermediate. If there is a liquid intermediate, it will cause some of the problems in the texture of these food products. The lyophilization process can maintain the texture of the many of these freeze-dried products. If you allow a product to evaporate by heating or something, that's going to change or cook the product. Whereas in this process, what you do, that you can quickly, say for an example, you can quickly freeze some vegetables, and in the frozen condition, you apply a vacuum. So all these water that is available in the iced condition in that freeze-dried food is going to evaporate directly from ice to the vapor form of water. There will be no liquid water intermediate, and that preserves the texture of those products, and that is called the lyophilization. So in one word, once again, the lyophilization is dehydration or freeze drying. So next, what we are looking at, the chemicals that is used for preservation, and the chemical agents are known as a GRAS by the FDA, and these are generally recognized as safe. And these can include many of the organic acids, sulfite, ethylene oxide gas, which is a sterilant as well, ethyl formate. The sodium nitrite in the meat products can be broken down into nitric acid, and this nitric acid can bind with the heme group of the myoglobin, and the myoglobin is the hemoglobin that is present in the muscle cells, and that can keep the meat red for a much longer time. Also, there is a problem with that. It also produces, which is called nitrosamides. These nitrosamides arguably could be a potential carcinogen, and that's one of the drawbacks. On the other hand, pH, say for an example, use of acids can inhibit microbial growth on meat because most of the bacteria that grows on the meat, they prefer a neutral pH. So acidic pH will prevent the growth of many microbes. So for example, acetic acid or lactic acid inhibit listeria. Organic acids usually at the level of 1 to 3% are used in meat. The poultry is preserved in 10% lactic acid and sodium lactate buffer, which is pH 3 before packaging. Sodium propionate is also another product that is used for the chemical-based preservation, and the activity of the sodium propionate actually increases at lower pH. Also, there is a common product which is a citric acid preservation, and this citric acid preservation is routinely used in home canning products when we make canned products at home as well, and these are readily available in supermarkets. So here is a major group of chemicals used in the food preservation, and you do not have to just memorize all of them. Just have an overall idea that what are the different types. Benzoic acid benzoate at 0.1% also prevent the growth of yeast and the molds, and you can see margarine, pickle, relishes, These are where the benzoic acid or benzoates are used. Dihydroacetic acid is a very interesting product which are used as a pesticide on strawberries without any. This is a product which is non-toxic, almost non-toxic as a pesticide, can be used in strawberries and squash. And you already know that ethylene propylene oxide, these are kind of sterilants and the fumigant for spices and the nuts. And these products are not present in the spices or nuts, but they are processed using these before they are sent into packaging. On the other hand, ethyl formate also inhibit the growth of yeast and the molds in dried fruit and nuts. And we talked about the paraben. This is a very common in bakery products, soft drink, pickles, and salad dressings. Then propionic acid, propionate, we just talked about that the activity of these increase in lower pH. This is also the inhibitor of ropey bread dough. Then sodium diacetate also prevent molds in the bread. Sodium nitrite, this is a very important one because it inhibits the Clostridia in cold cuts, hot dogs, or the sausages that we talked about in the last one. Very important product. Then sorbic acid or the sorbates, sulfur dioxide or sulfides, they can prevent the growth of insect and the microorganism in molasses, dried food, wine, lemon juice, etc. So another important thing which is not used in an industry still is a high hydrostatic pressure, which is also known as pascalization. This is an alternative way to preserve food and applies pressure to about 100 to 800 millipascals without significant changes in the temperature. And this is highly detrimental to cell membrane, and effective eliminating eukaryotic microbes means that mainly the protozoal contaminants can be reduced by this high hydrostatic pressure. It is not as effective as elimination of gram-positive microbes as well, but mainly the eukaryotic pathogens. There is no industry standard for HHP, and the HHP is not a mainstream process of food processing yet. The radiation is also known as radipartization from Nicholas Appert, who started this process of food preservation by different methods and use ionizing radiation like gamma radiation from a source of either cobalt-60 or a cesium-137 source. Ionizing radiation, gamma radiation, is extend shelf life of sterilized meat, seafood, fruits, and vegetables. And basically what they do that they will directly break the DNAs or the genetic material of the microbes. And they have excellent penetrating powers. The food is not needed to be heated nor rendered radioactive, which is a common myth, which is not true as well. In addition to break down the DNA of the microorganisms, so that is the main process by how the radiation works, it also kills microbes in moist food by producing these reactive oxygen species from water. And these ROS are the reactive oxygen species, which is produced by this ionizing radiation. And that's the reason that what you call is an ionizing radiation, such as gamma radiation. These, when produced in the environment of the microbes, can readily kill the microbes by oxidating the lipids as well as the proteins on their structure, outer structure. Then there are some microbial product-based inhibition, and bacteriocins are used. So bacteriocins are bactericidal proteins that is active against the relative species. And these are natural product of many bacteria which compete with each other to grow in a space. And some dissipate proton motive force of susceptible bacteria as well. And some form pores in the plasma membrane, some inhibit protein or RNA synthesis. So these are the different categories of bacteriocins and their mode of actions, 1, 2, and 3. For example, Lactococcus lactis produce a bacteriocin which is called a niacin. This is an amphiphilic molecule. So this amphiphilic molecule will just go and bind to the plasma membrane of other microbes close by, and then it will basically disrupt the membrane integrity and killing the microbes. Also used in low acid food, inactive Clostridium botulinum during canning process. Another microbial product-based inhibition is spraying of bacteriophages. So these bacteriophages are bacteria killing viruses. To kill Listeria monocytogenes, these bacteriophages which kill the Listeria are sprayed onto the ready-to-eat meats prior to packaging. So this is a very interesting application. On the other hand, the modified atmosphere packaging or MAP is also important. The gases in stored food affect the microbial growth. And the shrink wrap material and vacuum technology control the atmosphere in package. And this is a very, very interesting area that we see this all the time in all products in the supermarket. So the first of all, that first, some of the shrink wrap are impermeable to oxygen. So when there's impermeable to oxygen, there are many of the aerobes which grow on the surface of the food will not grow. So that's one of the important things. On the other hand, high CO2 content, like 60% of CO2 content, can be used to prevent in fungal growth. Even at 60% CO2, even if there is some oxygen in there, still it is inhibitory to the fungal growths. On the other hand, what happens that high oxygen content packaging produces this superoxide. Superoxides are similar to the reactive oxygen species, as we said, that which knocks off the electrons. And the superoxides, like O2 minuses, are also do have these unpaired electrons, and these anions inhibit the microbial growth by the same process, by oxidative damage to their structures. Now we are going to talk about the foodborne disease outbreaks. So we're going to talk about some of the major foodborne infection that substantial health economic consequences, origin, consequence of food intoxication, explain in these cases the foodborne disease outbreaks, cautionary tales for the government, regulatory agencies. We're going to look at what is aflatoxins, what is fumonizing. So here, this is just a historical perspective that about 48 million cases of food related to illness per year happens in the United States. And you can see that more than 100,000 hospitalization, 3,000 deaths per year due to this, and only 14 million attributed to known pathogens. And these pathogens are Salmonella, noroviruses, Campylobacter jejuni, E. coli. For example, this strain O15787 is an enterohemorrhagic strain and potentially life-threatening, could be Listeria monocytogenes. And these are some of the most important type of foodborne illness pathogen. The transmission, the main problem is the fecal-oral route is the key. And how this happens. This happens through the feces, finger food, fomites, and the flies. We easily can understand that the power of hand washing, that how these cross-contamination can be reduced. On the other hand, fomites, we have talked about that. The fomites are the objects which are non-living, which can harbor pathogens. So when the food handling is happening at that time, if it come in contact with a fomite, which is a non-living surface, which harbors some of these specific pathogens, that can lead to the transmission as well. And you can easily understand that flies, this is a concern, that the flies can sit on a surface which is very rich in microbial population that can carry just mechanically many of the microbes to food products. And there are two primary types of foodborne infections. One is the foodborne infections and the food intoxication, which is due to the production of the toxins by the pathogens. So let's take a look at these foodborne pathogens. So foodborne pathogen examples are the Listeria monocytogenes in daily meats, or see this enterohemorrhagic O15787 E. coli in undercooked hamburgers, spinach, lettuce. Many times we just see this, that a huge amount of recall will be from the supermarkets. You can see your textbook. This is a whole detail is written in there. And if you're interested, you can just take a look at that, all the different stories that is in there, which is essentially not needed for your quizzes or exam, but it gives you the stories of many of the over the period of time, that how many of these breakout of the foodborne infections happened and how it was traced back to where and how it happened. Some interesting read from your textbook. So food intoxication is the food in which the microbes have grown, and this produce symptoms shortly after the food is consumed, because growth of these disease-causing microorganisms is not required due to this. But this is just due to the contamination of the toxin. So here is an important thing, that E. coli 15787 produce something which is called a Shiga toxin. And this Shiga toxin gives you this enterohemorrhagic diarrhea or bloody diarrhea. But Shiga toxin is not considered as a foodborne intoxication, because usually in the patient, live E. coli 15787 is present. Only in the presence of E. coli, the Shiga toxin works. So usually if there is no infection of E. coli, Shiga toxin is not in the system. That's why this is considered as a foodborne infection, but not food intoxication. Whereas you can see that staphylococcal food poisoning, botulism, Clostridium perfringens food poisoning, Bacillus cereus food poisoning, these are all different types of food poisoning that produce toxins. If ingested, it can make the person sick. Say, for example, after eating in the picnics, if some started profusely vomiting, most of the time there are some staphylococcal enterotoxins are involved in that process. Home canning can produce these botulism. Clostridium perfringens toxin food poisoning can give serious types of stomach cramps as well, whereas Bacillus cereus toxins can produce also profuse vomiting. And usually this Bacillus cereus grows on starchy food like rice pastries, like those, and that can lead to these poisoning as well. So here, what we are looking at, this is the two important toxin, and these are the fungus-derived toxin. The first one is the aflatoxin, and these aflatoxins are carcinogens and produced in fungus-infected grains and the nut products. So aflatoxin is mainly produced by the Aspergillus flavus, and there are four basic chemical structures. You have this B1 and B2, and then you have this G1 and G2. So what it does mean that if you extract these particular aflatoxins, and after that, if you just separate them by chromatography, and after you purify them, if you just shine an ultraviolet light on them, these will B1, B2 will fluoresce blue, whereas these G1 and G2 will produce a green color. And on the other hand, what you are seeing that there are this M-type aflatoxin, and these are found in the milk of lactating animals if they do ingest the B-type aflatoxin. So this is an interesting type. So you can easily see naturally occurring, and this is occurring in the milk if the lactating animal, like a cow, ingested that aflatoxins. So as you can see from the aflatoxin structure, that these are planar compounds. So what they do, that they intercalate themselves in between the DNAs. And when they intercalate in between the DNA bases, the ATCGs, adenine, guanine, cytosine, and thymines, they cause frameshift mutations. And that's why they are called the carcinogens. And mainly what happens that they affect the liver first. That is the main route of exposure and can cause liver cancer and serious liver damage. If a person do have already some virus infection like hepatitis B, due to aflatoxin, they do have 30 times more chances to get cancer or hepatic cancer due to these aflatoxins. Unfortunately, what happens that depending upon the food that we take, there is a little amount of aflatoxins that is present in our food, and we naturally take it every day. But it depends upon the diet, and because they are naturally present in beer, cocoa, raisin, peanut butter, soybeans, meals, and depending upon the food that we take, say for example, in a typical European diet, the people do consume about 19 nanogram per day of aflatoxins, where in the Asian diet do consume about 103 nanogram per day of these aflatoxins. So at that level, it is really doesn't cause any apparent toxicity, but higher level can cause serious problems. The next one is known as the fumonizins, and here is the structure. There are two forms, which is known as the FB1 and FB2, and this is produced by the fungus known as Fusarium moniliforme, a fungal contaminant that could grow on improperly stored corn. And about 10 different type of fumonizins have been isolated, and these are strongly polar compounds, and that can cause disease in domestic animals and the humans. In horses, it can cause, which is known as leukoencephalomalacia. This is also known as blind staggers, which is fatal to the horses in about 2 or 3 days. It can cause also pulmonary edema in the pigs or esophageal cancer in the humans as well. So these fumonizins do inhibit an enzyme which is known as a ceramide synthetase, and that inhibits the formation of sphingolipids, which is an integral part of any plasma membrane. Also, the last one is the algal toxin. There are some algae, and some of this toxic algal blooms can also contaminate fish as well as shellfish. So the next portion we are looking at that how we detect the foodborne pathogen, and this requires the government industry cooperation, and we're taking a look at these procedures. So this is historically in 1905, this Upton Sinclair's novel basically revealed the horrors of meat packaging industry, and at that time the Federal Meat Inspection Act was passed. And then there were many over the period of time, many of the legislations were followed, and in 2011 there was a 2011 FDA Food Safety Modernization Act that has been passed. And this led to some of the basic process which test the food safety. The test must be very specific, it must be extremely sensitive, and it should be rapid or fast, and it should be a simple test. So these were the goal of these Food Safety Modernization Act, and there are several types of detection methods that were developed due to that. So one of the things that the culture technique means that you can isolate or swab the food samples and can grow the cultures. This is too slow and most likely to detect foodborne pathogen. This is probably not the fastest way to detect the pathogen. Immunological techniques are very sensitive, which can be ELISA, which is the enzyme-linked immunosorbent assay, or it could be a lateral flow test, which commonly we call as the rapid paper strip test. All of these are based on some of the immunological principles, or they can be used some of the biosensors. So how it actually works, that particularly if it is an immunological technique based, which basically look for a particular antigen which is present in a particular pathogen, and we use a corresponding antibody which can bind to that. On the other hand, the biosensor-based techniques can use a receptor, enzyme, or peptide which can detect a pathogen. And usually these biosensors are connected with a transducer, and when there is a positive match in between the biosensor and the corresponding microbe, there could be an optical, electrical, magnetic, or chemical signal which can give you a readout. The next is the molecular technique, where you can use the DNA or the RNA. This is also very sensitive and specific, and these include the PCR, which is the polymerase chain reaction, and there are many modifications of that, which is a real-time PCR. So in real-time PCR, the DNTPs, or deoxynucleotide triphosphates, are tagged with a fluorescence. And real-time, as the PCR reaction is going on, you can detect your microbe. Also, there is a process which is called a multiplex PCR. In a multiplex PCR, in the same reaction mixture, you can add multiple different primers, each specific for a specific pathogen, and that can lead to very specific type of readout, where for each specific pathogen, the PCR product will be of a different size, and that can lead to detection in a mixed population of foodborne pathogen. Then what we are looking at, that right here, the whole genome sequencing technique is a very popular technique, and mainly this is not carried out routinely, but when there is an outbreak, usually the industries and the government do work in collaboration to find out that what is the source and what if there is any new pathogen that is going around or not. And the reason that this whole genome sequencing is now become so popular, because now the sequencing technique or the next generation sequencing techniques is extremely fast and very powerful. So a few years back, there was a European strain of E. coli, which is known as the O104H4, that was detected, which is a novel strain that was detected within 1 month of the outbreak by this whole genome sequencing technique, which is very, very powerful. The WGS used for the routine surveillance of the certain foodborne pathogens and also analyze the virulence and antibiotic resistant evolution of those foodborne pathogens. So here what we are looking at is called the CDC's PulseNet, uses a pulse field gel electrophoresis to determine distinctive DNA pattern of each bacterial pathogen. And this enables the public health official link the pathogen associated with disease outbreaks in different parts of the world to a specific food source. So how do you use this particular product? So to do this pulse field gel electrophoresis, you isolate these foodborne pathogens, and these foodborne pathogens, once grown, the DNA is extracted from that, and the DNA is digested by some specific restriction endonucleases. Once these restriction endonucleases digest the whole of their genomic DNA, depending upon a specific pathogen, it will produce a characteristic pattern, which you can think about like a fingerprint pattern. So think about that. Suppose each of these lines are digested DNA bands, which is separated by the pulse field gel electrophoresis. I'm just giving an example. Suppose you take the first line. If you take this is just for your illustration purpose. Suppose you have the strain of E. coli 0157H7. You isolate that from the food product, or you have taken a food sample where you suspect that the pathogen is growing. So you isolate the pathogen and then you grow in the culture. This takes a little bit of time because this is leading to some outbreaks, those kind of things. So once you grow the pathogen, you extract the DNA from the pathogen, and suppose you digest that DNA by a restriction enzyme, which is say EcoR1. If you digest it with the EcoR1, if you just run that through a pulse field gel electrophoresis, you get a pattern like this. So once you get this pattern, think about that. This is like a DNA fingerprinting pattern. You upload this pattern into the CDC's PulseNet, and immediately it will match with the database, and it will tell you that that organism that you isolated is E. coli 0157H7. And that's how you can identify rapidly that what pathogen is the source of that contamination. Once it is confirmed, immediately if they're found in a particular set of food product, they are all recalled back. And this is the process. So next we are looking at the microbiology of the fermented food like beer, cheese, and much more. And I'm sure that you will probably enjoy this portion. So fermented products are the chemical changes in food brought about a microbial action. And these microbial fermentation used are lactic, propionic, and alcoholic fermentation, means lactic acid production, propionic acid production, and alcohol fermentation. So these are some of the main processes. So first we are looking at the different types of fermented milks. And this majority of the fermented milk products rely on the lactic acid bacteria, which uses the lactose from the milk and then produce the lactic acid from those products. And they are the genera of Lactobacillus, Lactococcus, Leuconostoc, Streptococcus. And these are known as firmicutes. This means that they have a gram-positive cell walls, and they tolerate acidic conditions as well. And they are non-spore forming, they are aerotolerant, means that they can tolerate air, but they do not need air or oxygen for their growth. And they're strictly fermentative as well. So let's take a look at how different types of fermented milks are produced. So all of these products or the fermented milks, they start with a product which is known as LAB, which is a short form of the lactic acid bacteria. So these lactic acid bacteria will contain all of these different, and then there are many other which can be added later as well. So on the other hand, there are some inherent problems out there that have to be careful in the industry. There is sometimes a contamination of bacteriophages which can kill the starter culture, and this is called a vexing problem, and that is also something that the industry which is producing this fermented product have to take care of. So for the production of the buttermilk or the sour cream, the Lactobacillus species or the Lactococcus lactis essentially do the lactic acid fermentation. After the initial lactic acid fermentation, by cooling, the growth of these two species are stopped. And at that time, there is another subspecies which is known as Lactococcus lactis diacetyl lactis. And this diacetyl lactase converts the carbon dioxide and milk citrate to diacetyl, and that gives the buttery flavor of this buttermilk. On the other hand, the yogurt is used at about 45 degrees Celsius to grow, and these bacteria, not very high temperature, but still they are considered as thermophilic bacteria. And there are lactobacillus, streptococcus thermophilus, and essentially the streptococcus thermophilus first mildly acidifies the milk, and then there is another species which is known as Lactobacillus bulgaricus, acidifies the yogurt more to convert more lactose to lactic acid. And then finally, the flavor is developed by the diacetyl and acetaldehyde formation. And sometimes the yogurts can be supplemented by some of the probiotics, other bacteria, but that is not the natural ingredient of the yogurt, but they do not have any role in fermentation, but they increase the food value of the yogurt. And then what we are looking at, that this kefir, they all originated in the Caucasus Mountain, but they are now found in all over the world in every supermarket. So this is a little bit foamy due to the carbon dioxide, and they do contain the 2% ethanol in there as well. But the starting product is a little bit different. The starting material do contain this kefir grain and casein, which is the milk proteins. So along with that, you have this yeast, lactic acid bacteria, acetic acid bacteria, and that produce by the fermentation this kefir milk. There is also another example. The Vili is a Finnish fermented milk, and this uses a filamentous fungi, which is known as Geotrichum candidum. This is basically a mold. This is a very specialized product, which is a smooth, velvety kind of textures that is given by this filamentous fungi. And along with that, some of the lactic acid bacteria are also produced, Vili, or the Finnish fermented milk. So here we are looking at the cheese production. So as you know, there are probably 2,000 or more different distinct varieties of cheese and representing about 20 general types. And the cheese is based on the texture, hardness, soft, semi-soft, hard, very hard. And they are all from the lactic acid fermentation. And sometimes molds are used to further enhance the flavor. So how it is done? So the milk may be pasteurized, and after to the milk, you add the lactic acid bacteria and you add the rennet mix. So the rennet mix do contain an enzyme predominantly, which is called the rennin. This is an enzyme which helps the animal to digest the milk. And this produce what is called, in about 30 minutes, it produces what is called the curd or the cheese curd. And this is the fermentation followed by the fermentation of the cheese curd, and the pH drops due to lactic acid fermentation to about 6.4. So once the pH drops to about that, take the curd and then cut into small pieces and then heat it about 38 degrees Celsius to separate the whey, and the liquid part or the whey is removed. At that time, you will get something like this product, and constantly these slabs will be turned around. And in this process, more and more of the liquid part will come out, which is the whey. And at that time, this flipping action will also reduce the pH even further to pH 5.1, 5.5. And this will form, which is called the matting of the curd, or kneaded together, or reach pH about 5.5 or 5.1. At that time, they are cut into small pieces, and they can be processed even further. They can be added with salt, different flavoring agents, or say placed in brine, say for example, mozzarella cheeses. And then also it can undergo ripening by microbial actions. And sometimes there are some cheeses like say blue cheese or Roquefort cheese. There are some of this particular mold that has been used. So in the blue cheese, you see that this bluish green, that is basically a Penicillium species, some kind of, in a Roquefort cheese, this is Penicillium roqueforti, that gives that unique flavor of these cheeses. So these are the process of cheese production. So here is a big list of meat and fish often modified by fermentation. This is very self-explanatory, and you can easily see that there are some specific ones. Juice is a fermentation of fresh fish, rice, and vegetable. And here you can see that katsuobushi is the fermentation of tuna. Most of these fermented products use the bacteria lactobacillus, but there are some examples of they use a different microbe, which is, say, for katsuobushi, which is a fermentation of tuna. They use this Aspergillus glaucus, which is a mold, instead of the lactobacillus. These are very self-explanatory. Just take a look just for your idea. Next, we are looking at the wines and the champagne production, and the wine production is known as enology. So initially, we crush the grapes and then you separate the stored liquid, and the fresh must be treated with the sulfur dioxide fumigant, and then Saccharomyces cerevisiae or ellipsoideus added to the consistent result and fermented for about 3 to 5 days. So wine is contain about 10 to 14% alcohol, and as it increases the alcohol concentration at about that percentage of alcohol, it will automatically kill the yeast that produces it. So this process is very self-explanatory, so you can easily take a look at this. But the main thing that is important, that the grapes do have this malic acid and the tartaric acid. And if the levels of the malic acid and the tartaric acid do not decrease, it will give a sour mouthfeel, which is not very pleasant. To decrease the acidity, sometimes Leuconostoc plantarum, Hilgardii, or Leuconostoc brevis, they are added, and they convert this malic acid to lactic acid. And this process is known as malolactic fermentation. And this results in deacidification and flavor stability. And there are some possibility that they also produce some of the bacteriocins, which also preserve the wine. Once the acidity is reduced from this settling vat, all the excess yeast is removed, they are packaged, and this is aged, which is called the bouquet formation of the wine. Then it is bottled, and racking also ages the wine as well. So the dry wines do not have any sugar present or less amount of sugar present once the fermentation is over, whereas the sweet wines do contain sugar after even fermentation. And racking, storing wine while it ages, removes the sediment produced during fermentation, and when fermentation continues in the bottle, CO2 bubbles form, resulting in sparkling wines or the champagne. And there is an interesting way that how this champagne is bottled. After the champagnes are bottled, they are turned upside down, and the neck is actually frozen and uncorked, and the sediment which settles at the bottom of these champagne bottles are removed. They are filled with the fresh champagne from the bottom, and then they are sealed back as well. So very interesting way that how the champagnes are sealed and how the sediments are removed gives its very clear appearance. So the next is that how the beers and the ales are made. These are starting material is cereal grains like a barley, wheat, rice used for the fermentation. And the first thing is the malt, which is the germinated barley grains having then activated hydrolyzing enzymes. So once this barley is moistened and germinated, then they are dried and they're crushed, and they are put in a mashing container. And in the mashing container, the further enzymatic activity release the maltose, the dextrin, and the proteins. And at that time, the hops are added. The hops are from a dried flower of Humulus lupulus. And once those hops are added, then in a brew kettle, the temperature is increased. And at that time, the spoilage organisms are inhibited, enzymes are inactivated, and the flavoring from the hops causes also the clarification. So next, after the brew kettle, the hops are removed. And at this point, the product which is formed is known as wort. So this wort is the product which is fermented. At that time, the yeast is added and the fermentation goes on. Once the fermentation goes on, they are stored, which is called as a lagering process. And that's for the final flavor development. And then it can be packaged. Sometimes there are natural carbonation inside the bottle that goes on, or sometimes additional carbon dioxide is added into the bottle or the cans. because sometimes some of the beer which undergoes pasteurization will not produce this carbon dioxide in the bottle or the can. Those are added with the carbon dioxide from an outside source. So mainly there are two ways that the beers are produced. One uses a strain, which is the Saccharomyces pastorianus, and about 7 to 12 days of fermentation brings the pH to about 4.1, 4.2. And sometimes most of the beers use this particular genus and the species, Saccharomyces pastorianus. Some of the yeast, which is a top yeast or the Cerevisiae, that drops the pH to 3.8, and these are used to produce the ales. So that's the difference between the normal beers and the ales. So here we are looking at some of the terminology. The wort is the product of the initial processing, and the wort is pitched or inoculated with the desired yeast. The bottom yeast are used for the production of beers, whereas the top yeast, like Saccharomyces cerevisiae, are used for the production of the ales. And the freshly fermented beers are aged or lagered, and sometimes carbon dioxide can be added for the bottling, and beer can also be pasteurized or sterilized by filtration process as well. On the other hand, for the distilled spirit, is basically an extension of the beer making process, but it starts with something which is called a sour mash, and this sour mash is inoculated with homolactic bacteria, which even lowers the pH. So the sour mash is the liquid fermented grain from a previous batch, and the sour mash is inoculated by a particular lactobacillus, which is Lactobacillus delbrueckii or subspecies bulgaricus, which lowers the pH to about 3.8 in about 10 hours. And this inhibits development of undesirable microbes, and then it is boiled and the volatile components are condensed for higher alcohol content than beer. And there are several types of beer. Say, for example, rye whiskey will contain 51% of the rye. Bourbon is from the corn. Scotch is from barley. On the other hand, gin and vodkas can be made from a different number of grains as well. In the gin particularly, sometimes there are some juniper berries and other flavoring agents are added to give its characteristic taste and flavor. Bread is one of the oldest human food, and it involves the addition of the baker's yeast or the Saccharomyces cerevisiae under aerobic condition. And it maximizes the CO2 production, which leavens the bread and the rising of the bread. And there are other microbes that can be used, say, for example, sourdough bread, and the breads can easily spoiled by bacillus species that produce the ropiness of the bread. On the other hand, we have already seen that being a very good source of carbohydrate, many fungus like Penicillium, the green mold, can grow on the bread, or sometimes a black mold, which is the mucor species, can grow on the bread as well. Other couple of fermented fruit from around the world. Sufu is for the fermentation of the tofu, and the tofu is produced by a fungus, which is a mucor or actinomucor. Sauerkraut or sour cabbage from the shredded cabbage. This is mainly the fermentation by leuconostoc and lactobacillus. So the pickling process is basically the soluble carbohydrates that is present from the cucumber. These are converted into the lactic acid. And also along with that, sometimes the gram-positive leuconostoc, lactobacillus, enterococcus faecalis, pediococcus, many different types of bacteria can be present in the pickle as well for its maturity. This is an interesting product which is called the silage, and the silage is from grass, chopped corn, and other fresh animal feeds. And this is a fermented product, and this is usually used during the winter months to feed the animal. At that time, they will not be able to graze in the region where there is a huge amount of snowfall. And this is a form that they are used in the winter months. Also, along with that, you can just take a look at your textbook. You have a huge chart where you can see that your coffee is a fermented product, gari, which is a cassava fermentation in West Africa, and kimchi from the Korea, miso that we use, the miso soups, very popular in Japanese cuisine. Then your soy sauce, fish sauce, these are all other fermented fruit from around the world that we enjoy. It's a very pleasant chapter in the sense that although we talked about some of the spoilage of the food products and how to detect the microorganism on the food, but there are many fermented products that we enjoy, and you can just really take a look at that, how they are produced. And now from here, you can go on and look at the process of certain products, increase your knowledge even farther that you particularly like. That will be the end of chapter 41.